Scientists built a synthetic cell from non-living components and watched it feed, copy its genome and reproduce for 5 generations. They call it SpudCell. The researchers began with a liposome, a microscopic compartment enclosed by a lipid bilayer similar to a cell membrane. Inside it, they placed a 90 kilobase genome divided across 7 circular DNA molecules along with purified molecular machinery that could read the genome, produce proteins and copy the DNA. But there was a major problem. The synthetic cell could not make enough of its own components to keep functioning. It needed a way to acquire fresh material. So the researchers created much smaller feeder liposomes carrying new membrane lipids, ribosomes, enzymes and other molecules required for protein production and genome replication. SpudCell was designed to produce a protein in its membrane that allowed it to recognize and fuse with these feeders. When the membranes fused, their contents mixed. The synthetic cell received fresh molecular machinery while the additional lipids expanded its membrane. That was its version of feeding. After feeding, the genome was copied, the compartment grew, and the researchers divided it to produce daughter cells. They repeated this cycle for 5 generations and found that some descendants were still copying DNA and producing proteins. Then they added competition. One group of SpudCells carried a genetic change that made them produce more of the membrane protein needed for feeding. These cells fused with more feeders, grew more, and produced more daughter cells. SpudCell is still far from an independent living organism. It cannot make its own ribosomes, its metabolism is very limited, and researchers still have to supply the feeders and assist parts of the reproduction process. The 5-generation experiment specifically used mechanical division, although the team also demonstrated a separate method in which proteins produced by the synthetic cell help trigger division. The study is also currently a preprint, but it gives scientists a controlled system for asking a fundamental question. What is the minimum combination of components needed for a chemical system to feed, reproduce, and undergo selection? Follow for more interesting science content.